Wait, what?
No, it's basically weird. It would all be the same thing. I'm not three. Right, I'm saying you. Are you guys all close? Oh. God. Oh my God, I'm so. Wait, what do you know why? You're the one, yeah. I thought you were gonna be like 90. I'm 99. I'm 25. We are?
No, you're not.
You're someone else.
All right, good morning, everyone. Let's go ahead and get started because we have a few things to do today. As promised, we're going to go over worksheet 3, and so we'll go through each question one by one. All I ask is that I get some participation from more people this time around and have, more of you guys answering these questions than the same people. So, yeah, who wants to tackle number 1? Why is a virus considered a microbe but is not living? Yeah.
They can't replicate by themselves. Like they need those cells to do it.
Yep, exactly. They can't replicate independently, and they utilize the cell's machinery in order to reproduce. So they take advantage of the host. So, simple. Number 2, what are the 2 components that all viruses have? This is something, yeah, right.
Capsid and genome.
Yep, exactly. So a nucleic acid genome, either DNA or RNA, and then a capsid that is made of proteins. All right, compare and contrast the forms of tailed, icosahedral, and filamentous viruses. There's 20 questions here, and there's about 20 of you here, so I expect at least one person to answer each question. Yeah, Sarah.
So filamentous, the capsid has a long tube of protein. They vary in length, and it depends on the gene size. And then it has a phage. And then for tail, it has a head that's pressure packed with US DNA, and then a tail sheath covered in virus. And then I couldn't find the ISO.
Yeah, so the icosahedral, I kind of, talked about that briefly. Basically, it's the tail. It's like a tailed phage, but without the tail. So it's tailless. So exactly, I just wrote down that it's same as the tail, but without the tail. So it's just the icosahedral head that the tailed virus has. So it doesn't have the tail sheath and the legs, the tail fibers at the end. Yep. So the icosahedral is just the head part of the tailed viruses. So the tailed ones, sometimes they have that elaborate delivery device, which gives them that tail, that sheath, and then also the tail fibers at the end. Icosahedral viruses just have the head. They don't have that elaborate delivery device, the tail sheath and the tail fibers. So it's just the top part of the tailed, or it's tailless. Does that make sense? All right, any questions on those? Just some structures of viruses. All right. Number 4, what's a bacteriophage? And explain how a pure isolate of a virus can be obtained and how do these procedures differ from those used for isolating bacteria? So answer what you can from this. We can kind of go through it one by one. So what is a bacteriophage? Yeah. Exactly, bacterial eaters. And then how do we get a pure culture of a virus? How do we see viruses in culture, on a plate, or in media? Yeah.
If they're host specific, by growing a bacteria, host bacteria, you can cultivate viruses.
Yep. So for those of you who may have not heard that, basically we grow them on a bacterial lawn. So we grow the virus with the bacteria that it's supposed to eat or infect, and then it will form these plaques. So the difference is basically between this and the next part of the question where I ask how is it different from isolating bacterial colonies. In bacterial colonies, just like with your street plating, you guys are getting those individual colonies. Those are thousands of cells of the same bacteria that was on the plate, so they form a colony. Whereas on a plaque, you kind of see the reverse. You see a plaque, which is that zone of inhibition, the area where the cells have lysed the bacteria. Instead of seeing a colony, you kind of see like the negative inverse effect of it, where you just see that hollow plaque, so that hole in the lawn, essentially. Does that make sense? You need the bacteria there in order for the virus to eat away at them and then create that hole. And so with that, in that plaque region, the very, very, very center of it is where one viral particle infected one bacterial particle, and then it was able to utilize the bacteria and reproduce and kill more and more and more. So it basically, when we see plaques, it's undergoing the lytic cycle. So it's killing all of the bacteria around it, and therefore you get that perfect circle with a certain diameter depending on how much the virus was able to eat away the bacteria. So then that gets into bacterial susceptibility and that kind of thing, but that's generally the concept. And so, yeah, what's one key contribution of viruses to natural ecosystems like our gut microbiome? Yeah.
It limits the number of bacteria present.
Yep, exactly. Yeah, so that's one thing. And so what are gut viruses called? Yeah.
Coleophages?
Yep, colephages. Coli comes from the term colon, so it technically refers to the intestinal tract. And so, yep, the colephages, they modulate different things like digestion, our immune system, our mental health. And so that's all areas of research still that's being studied. But yeah, exactly. Phage can limit the bacterial numbers to levels that our immune system can tolerate because we have bacteria in our guts as well. And then they can also attack biofilms of bacteria. That's another one. All right, any questions here so far?
Are you recording this?
Mm-hmm. Yeah. Okay, so explain the steps of a bacteriophage starting an infection. So there's two main things. One is recognizing the host cell, and then the second thing is actually injecting its DNA or its genome. So how does it start an infection? Yeah.
So the first part happens by the contact or attachment that's mediated by the cell surface receptors. And then it said usually that the proteins are specific to the host species that they bind to. Mm-hmm. And then the second step is like injecting their genome. So usually it occurs through a cell envelope, but there's like 3 sub-steps. Mm-hmm. The first is the sheath contracting, and then it brings the head closer to the surface DNA. The second is the pressure, and it like spools out the DNA into the cell. And then the third is like the ghost capsid, which is like just attached after on the outside.
Yeah, exactly. So it's important to kind of keep some of these things straight in your head when I ask things about like starting an infection. You know, that's literally just the attachment at the beginning, the contact with the host cell, and then how it injects its genome, and that's exactly right. And so what are some different things that can serve as receptors for the phage on the bacteria? Yeah, flagella is a good one. There's also other components of the peptidoglycan and cell membrane, like outer membrane proteins, LPS components, that's that lipopolysaccharide transport channel, so proteins that are embedded in the cell membrane, as well as the flagella or pili on bacteria can be phage receptors, can serve as molecules for phage receptors. Okay. Yeah, and so basically the cool, textbook example that I gave in bacteriophage injecting its genome is that tailed virus. So it has the head, it has the sheath, it has the legs, the tail fibers, and basically, kind of just reiterating what was said, the sheath has to contract and bring the head all the way down, so it kind of squats and poops out the genome, if you remember that analogy that I gave. And so there's that DNA in the head is very, very pressure packed. So when it squats down, that pressure kind of just makes it spool into the cell. And so that's how it can get in. And then you have that capsid protein, the head, that we call now a ghost because the DNA has been injected, but that part remains on the outside of the cell. All right. Compare and contrast 3 different life cycles of phage, so we can go one by one here. Let's talk about the lytic cycle and try to parse out some of the differences. Yeah.
I said that one leads to death of the cell.
Yep, exactly. It does lead to death of the cell, but so let's kind of go through this a little bit. So first you have that host recognition and attachment, and then you have entry of the genome. So basically, the cell, the virus gets into the host cell and it has access to its cell machinery. And so because it has access to the bacterial cell machinery, it uses those to make its own capsid proteins and start to make its own phage components. And so those are expressed and start to assemble into progeny phage. So you end up getting the phage DNA inside of the cell, and then the phage can utilize the bacterial cell machinery to make its own proteins and make its own capsid head and other stuff, and then it can replicate that DNA and put it into those components, and it's all still happening inside the cell. And then once it does that and you have enough of the progeny phage, they exit the cell, kill it, and you get transmission. So those progeny phage also are exiting too, and then they go and look for other cells to infect. So it's a kill, kill, kill cycle. It's important to know that the phage is replicating inside and it's using the bacterial cell machinery for that one. Okay, lysogenic. Yeah.
That they either embed into the host cell's DNA or they stay in their own circulation and lysogenize the cell. And then I just say that they stay comfortable.
Yeah, exactly. So in the lysogenic phase, this is really a temperate phase. It's never always going to be just the lysogenic phase. Okay, so basically these are temperate phage that are quiescent or just kind of happy, and they are prophage at this step. So that's important to know. And what a prophage is it basically integrates the DNA into the host DNA. So like Juliana said, it could be as a circular chromosome, or it could just, embed itself. And so it's important to know that now it's existing as a prophage. So its DNA is just integrated. And so in this one, I guess one question that I should ask you guys is, when the virus is reproducing, is it using the host cell machinery and being active on its own and trying to reproduce, or is it just taking advantage of what this host cell is doing? Yeah, exactly. It's basically, it's not utilizing the host cell machinery on its own to just reproduce itself. It's basically just taking advantage of the host bacterial cell, just replicating, and it's also doing it too because it integrated its DNA. And then the other important thing to remember is that it can reactivate to become lytic, so it's in this temperate phage phase. The slow release cycle.
I said unlike the lysogenic and lytic cycle, the slow release doesn't have no cell lysing, and also the reproduction or the replication of the host cells is continuous.
Yep, so exactly. So basically, the phage produces particles inside of the cell. So I gave you the example of the filamentous phage where basically it injected its genome, which is just the initiation of infection for all of them. They inject their genome, and so then the phage will start to produce its particles inside the cell, but it doesn't lyse the cell. So the host is able to continue to reproduce, but it does so more slowly than uninfected cells. Anybody know what the reason is for the host still reproducing but a lot more slowly? Yeah.
Have to reproduce to stay like alive so that the phage can continue using it.
Yeah.
It's slower because the phage is using all of it.
Bingo. Yeah, exactly. So yeah, the host is still reproducing, so it doesn't recognize that the phage is kind of hiding out in there, and so now the phage is using its resources. So unlike the lysogenic, it's using the cell machinery of the bacterial cell because now the host is reproducing very slowly. But like the lysogenic form, it is also not killing the cell. It's not lysing the cell. So basically it infects. The phage will form a double-stranded circle in the case that I showed you. And so that DNA is replicated and the phages can assemble and exit the cell without lysing it. So the cell can continually make phage while the cell population is still growing. So in the slow release one, there's some similarities to the lytic, some to the lysogenic, so it's important to kind of keep those straight, where it's exiting the cell just like in the lytic cycle, but it's not lysing it, it's not killing it. What are sigma factors and what role do they play in gene expression? Is everybody good on viruses and everything before we move on to the next part? Okay. What are sigma factors? What do they play? What role? Is your hand up? Sorry. Yeah, Henry, and then I'll get you next.
I said sigma factors are proteins, and their role they play is they guide RNA polymerase to a specific DNA.
Yep, exactly. And what part of transcription are they necessary for?
Initiation.
Yep, exactly. So sigma factors are proteins. They are required for only initiation of transcription, and they bind the polymerase first and help it detect the promoter. And so it's important to know that a single bacterial species can make several different sigma factors because of that changing environment that it's going to be in. So it helps the cell to readjust its physiology, so it has roles in that as well. So if I ask you, a question on an exam about the role of sigma factors, and it's like A through E, and E is all of the above, that's one of those things where it would have multiple things. I would probably say something like it's a protein, and it's required for initiation, and it helps bring the polymerase to the promoter, and then it also, helps readjust the cell's physiology. So those all are interconnected roles, but that's kind of the type of questions I'll be picking from. So kind of merging all those concepts together. All right. Describe the 3 phases of transcription. I kind of wanted you guys to put this in writing because there's also 3 phases of translation. There's a lot of similar terminology, so I kind of want you guys to keep the concepts straight. So who wants to do this one? Yeah.
So first was initiation, where the RNA polymerase coenzyme binds to the promoter, and then elongation, where the RNA chain extends from the 3' RNA polymerase detaches from the DNA.
Yep, exactly, yeah. And what is that holoenzyme, the RNA polymerase holoenzyme? What does it consist of? Yeah, exactly. So when we talk about the holoenzyme, we're talking about the actual RNA polymerase with the sigma factor attached so that it can guide it to the promoter. But everything else that Erica said was right. What are the two transcription termination signals? So in termination, the RNA polymerase detaches from the DNA and the transcript is completed and released. You get that mRNA. So what are two different termination signals and how do they differ?
There's RHO dependent, which relies on the protein RHO, and then there's a strong pause site at the 3' end of the mRNA, and then RHO independent, which looks at the GC rich region of the RNA.
Yep, exactly. So rho dependent, basically it binds the RNA transcript and it kind of like pulls itself closer up to where that pause site is. So it relies on rho pulling itself and the pause site. Once it reaches there and it contacts the polymerase at the pause site, then the rho protein has this helicase activity. Basically what it does is it causes it to terminate. So that's what you should know. And rho independent, it doesn't require rho, hence the name, but it does require a GC rich region of the RNA. And then that also creates these uridine residues. And so that weakens the stability and the interaction of it, and so it makes the RNA polymerase fall off. All right. 11. So describe the 3 stages of translation or protein synthesis. And why is a ribosome called a ribozyme?
Initiation, it brings 2 ribosomal subunits, placing the first amino acid at position, and then during elongation, it adds an amino as directed as the mRNA transcript, and then termination, it releases the completed protein and recycles the ribosomal subunits. And then I said it was a ribozyme because the ribosome makes a peptide bond that stitches the aminos using peptidyl transferase.
Yep, exactly. So the ribosome essentially makes these peptide bonds called peptidyl transferase, and so that gives it enzymatic activity. So it's an enzyme as well as a ribosome, given the term ribozyme. And so in initiation, you said it brings the 2 ribosomal subunits together. What are those ribosomal subunits and what do they make? Yeah. Yes, the 30S and the 50S to make the 70S. All right. So tRNAs and RNAs, I said, were much more stable than mRNA. How do we determine the stability of RNA, and what do they have that make them less susceptible to degradation? So what makes them more stable? This is a little bit of a more difficult one.
The half-life determines their stability, and they have bases that are less susceptible to degradation.
Exactly, yeah. So we determine stability of RNA molecules by looking at their half-life, and so basically the time in which it can degrade its components by half. And so what makes them less susceptible is they have these modified bases that protect them from degradation from RNases. So yeah. All right, describe the 3 functional regions of a transfer RNA molecule, tRNA.
The anticodon, which hydrogen bonds with the mRNA codon specifying an amino acid. 3' acceptor binds the amino acid, and the side loops are recognized by enzymes that match tRNA to the proper amino acid.
Yeah, exactly. So the tRNA molecule basically has to be charged, and so it's that anticodon portion, that hydrogen bonds with the mRNA molecule or mRNA codon that has the amino acid that's going to charge it. And then, yeah, exactly, the 3' acceptor end that binds the amino acid, and then those side loops that match it to the proper amino acid. So, and the whole purpose of the tRNA molecule having these 3 functional regions is so that it can get charged with the right amino acid. Okay, and so during translation, a tRNA molecule has to move through 3 sites in the ribosome. So what are these binding sites in the ribosome? Peach.
The E site, which is the acceptor site, the P site, which is the peptidyl tRNA site, and the E site, which is the peptide site.
Yeah. So the A site is the acceptor site, and that binds the incoming amino acyl tRNA. And what is the amino acyl tRNA? What have I been calling it? Charged tRNA, exactly. And then the P site harbors the tRNA with the growing polypeptide chain. And then the E site's the exit site, so it binds a tRNA that's recently stripped of its polypeptide. So if you think about it, the tRNA is a transfer RNA, it's transferring that amino acid, so it has to get charged with it. It takes it to the ribosome accepts it, and then that tRNA no longer has an amino acid, so it's no longer charged, so it has to exit and then go find another amino acid to bring to the ribosome. Does that make sense? Okay. What is a polysome and why are they important for the cell? Yeah.
There are multiple ribosomes on an mRNA, and they protect the mRNA from RNases and speed the production of proteins from single transcripts.
Yep. So once a ribosome begins translating the mRNA, another ribosome can immediately jump on to the ribosome binding site. And so therefore, the mRNA can have many ribosomes moving along its length at the same time. And so this multi-ribosome structure is the polysome. And so, yeah, exactly. They protect the mRNA from mRNases and speed up the production of a protein. So this is one difference between prokaryotes and eukaryotes, where in eukaryotes, once again, translation and transcription are not coupled and things have to happen one at a time. They can't happen simultaneously, whereas you can have multiple ribosomes jumping on and doing things in prokaryotes. All right, let's go through some antibiotics that inhibit protein synthesis in bacteria and their differences. So streptomycin wants to tell me about that one. Yeah.
Streptomycin binds the A site, it acts for improper codon matching,
Yep. Important things there are that it binds the A site. You still get translation, but it results in the wrong protein sequence. Tetracycline.
It binds near the A site and then prevents and becomes settled with the mRNA from binding to the A site.
Yep. It binds near the A site and prevents the charged tRNA from binding the A site of the ribosome. Chloramphenicol.
Binds near the P site to inhibit peptide bond formation.
Yep, so it actually binds the P site. And so, yeah, it inhibits peptide bond formation. So it doesn't bind near it, it binds the P site. And then erythromycin.
Binds a peptidyltransferase site and alters the peptide bond.
Yep, so it alters it. So remember, the peptidyltransferase is the ribozyme, the site of the ribosome that is catalytic. So if it binds it, it's going to alter the peptide bond formation. All right, so this is kind of more of like a list. I didn't need you guys to, know any specific details here, but what are some ways that proteins are processed after translation, so post-translational modifications? Yeah.
I said folding and cleavage.
Yeah, so informal methionine cleavage, remember that methionine that is the start codon, cleaving that off is one way. You can also have adenylation, acetylation, lipidation, phosphorylation, methylation, glycosylation, all the ations. So, and a lot of different ways to modify proteins, but we didn't go into all of that. Essentially what that means is they're adding certain groups to the protein. What are proteins that help fold other proteins called, and give an example?
You said they're called chaperone proteins, and then there's DNA chaperone protein, which is like, I think, like a palm that carries it from the cytoplasm to the membrane, like, so it doesn't get degraded.
Yep, exactly. Basically is like the palm of your hand, and it clamps down on the polypeptide to assist in folding. And then the second one was the GRO-ES complex. So I can go through that one. So basically, the protein that's going to be folded fits inside of the stacked barrel. The stacked barrel is the GRO-EL, and GRO-ES acts as the cap. And so you basically have this cap coming on. The unfolded protein comes in, cap comes on, folded protein comes out, and the cap is off, and then another unfolded protein. So it's this two-part complex. You have the GRO-EL and the GRO-ES. And so I told you that ATP hydrolysis is used in both, but what is the ATP hydrolysis really important for? Is it for folding the protein, or is it just from releasing the folded protein from the chaperone? Yeah, so it's electrostatic interactions that forces it to do the folding, but the ATP hydrolysis is involved, but it's just to release the protein from the chaperone. So that's important to know. Okay, number 19, what are protein degrading machines called, and what is a circumstance in which the cell knows to degrade a protein?
It is proteasome, protease, which is the misfolded proteins are recognized by the protein product due to the hydrophobic regions being exposed.
Yep, exactly. Proteasomes are protein degrading machines, and what they have is they contain these degradation signals called degrons. And so these degradation signals are able to find proteins that are damaged or misfolded. And one example in which the cell knows to degrade a protein is if you have a misfolded protein that has hydrophobic regions that are exposed to the extracellular space. Okay. And then lastly, some proteins are destined to be bound within the membrane, and so this is a protein traffic control mechanism. So describe it in the context of co-translational export in E. coli. I want to do this one. Anything else?
I'm not really sure about this one, but
It's okay.
the ribosome is paralyzed by the SRP, and then translation recommences, and then the transmembrane process is
Yeah.
the transmembrane pieces are released into the membrane.
Yeah, so I'll go through that. So thank you. proteins that are meant for the cell membrane have these tags on them. These tags are these hydrophobic regions. Because remember, the cell membrane is the phospholipid bilayer with the heads and the tails. The inner part is the hydrophobic tails. And so these proteins that are meant to go there, they have these hydrophobic regions because they have to be embedded into the membrane where the hydrophobic regions are. And so they have these tags that are bound by this complex called the signal recognition particle, or the SRP. And so the SRP basically paralyzes the ribosome, so it doesn't continue translating proteins until it encounters another protein in the membrane that tells it that it can start translation again. And then that translated peptide is fed into this other complex. But essentially what you need to know is that these proteins that are meant for the cell membrane, they have these hydrophobic regions already because those hydrophobic regions need to be embedded into the cell membrane. But remember, I just told you right before that misfolded proteins that have the hydrophobic regions that are exposed can get degraded by proteasomes. And so that's why translation has to stop. And so there's that SRP, that signal recognition particle, that tells it to stop translating so it doesn't get thrown out of the cell and degraded, so it doesn't think it's misfolded. It's actually meant to go somewhere else. So there's that SRP protein that helps to take it to the area where it's supposed to be, and then it can continue translating and it meshes in with the membrane. Does that help a little? Okay. Awesome. So what do you guys think? Do you think that doing this is helpful in reiterating concepts?
Yeah.
Okay, cool. So I guess we can move on and talk about some things in lecture 9. So I don't know, maybe we'll get through it all. If not, we can continue on Monday. But today because bacteria, like I've said before, are constantly going into these different environments, it has to know when it needs to alter its physiology and its gene expression profile. So there's different ways to regulate those molecules. So some suggested chapter readings. Today I plan on going through gene expression and operon control, and next week quorum sensing, so we'll see what we get through today. Just a quick overview. I'm just going to skip just to save a little bit of time. And so, like I said, there are some genes that are tailored for specific environments, but not for others. And a bacterial genome encodes thousands of different proteins for many different environments. So, to successfully compete with others, the bacteria won't waste energy making unneeded proteins. And so there are elegant mechanisms to control gene expression at various levels. So we've talked about transcription and translation, and so today we'll kind of talk a little bit about how there can be some modifications to control gene expression. And gene expression can be controlled at the transcriptional level or at the translational level, and then there are these post-translational levels as well. So we're going to focus a little bit on the post-translational for a second, and so there are a number of ways in which you can modify the peptide to activate it, degrade it, or sequester the regulatory proteins. And so what we'll talk about today is that these post-translational mechanisms can act as activators or repressors for operons. So just another little, you don't have to know all of this. This is kind of just reiterating the concept that there are many levels of control at the gene expression level. You can alter the DNA sequence. So basically you can activate or disable a gene by flipping it, and it turns off the expression in some cases. You can control transcription by repressors and activators and sigma factors and other things. You can basically control the mRNA stability, so there are RNases in the environment that can degrade and regulate the mRNA levels. And so at the translational level, they can basically sequester the ribosome binding site, which is the RBS, in the mRNA transcript. And so this is just a summary of different examples. We talked about post-translational control with cleavage, phosphorylation, and methylation. So the important thing here is that control at the DNA sequence level is the most drastic and the least reversible. So if you have a mutation in the DNA sequence, it's going to be really hard to revert it back to its native state because the genetic code, is being disrupted. And so at the protein level, control at the protein level is more rapid and most reversible because the genetic code has been maintained already in the DNA at the protein level. So cells use different mechanisms to sense and respond to conditions within or outside the cell. So a microbe needs to detect whether it's floating in a pond, whether it's in your GI tract, etc. And so it has to sense that environment. And so once it senses that environment, the cell can then change the gene sets that it expresses to meet the needs for survival in that particular environment. And this is largely controlled by these regulatory proteins that specifically bind to compounds that are provided by the environment. We call these ligands. So just to give you an example, different regulatory proteins bind different sets of ligands. And so you could have a regulatory protein that binds a carbohydrate and then alerts the cell that there's a carbon source available. And so once that ligand is bound, it can then alter the ability of the regulatory protein to latch on to the DNA regulatory sequence, and then those proteins can then be made. So just a little general concept, but I'll definitely get into this a little bit more to make more sense. There are two counteracting forms of gene regulation, and we have repressors and we have activators. And so repressors bind the regulatory sequence in the DNA, so that's this sequence here in green, and prevents transcription of the target genes. And so many of these repressors can repress without binding a ligand, but some of them have to first bind a small ligand. Activators, on the other hand, bind the regulatory sequence in DNA and stimulate transcription of the target genes. But most of the activators actually have to bind a small ligand first. So let's talk a little bit about regulation of transcription by repressor proteins. So typically repression can happen in two general ways depending on the mechanism or scenario. So let's talk a little bit about scenario one here. You have your repressor protein here in red. And so basically this will bind the DNA sequence, and at this point it doesn't require a ligand, so it can just bind, and it represses prevents transcription. Target gene is not expressed. And so the relief from repression requires a specific ligand, which here we're calling an inducer. So basically, in order to relieve this repression and remove this repressor protein, this ligand has to bind to it, it removes it, and then you can induce transcription. So because that ligand is required, the increased expression of that target gene is now called induction. It's not activation, it's induction, because you have an inducer. Now, in scenario two, so basically you have this repressor protein that on its own, this orange molecule, it doesn't bind the DNA really well. It's pretty weak, so it needs this ligand, which is its co-repressor. And so both of these together can then bind the DNA. And so then that ligand, remember I said, is usually something from the environment. So once that ligand disappears from the environment and from the cell, it's no longer available to bind the repressor protein, and so the repressor protein can dissociate, and then the target gene is expressed, and so this is called de-repression. So where you get removal of the ligand and then induction of transcription. So in scenario one, you needed the ligand to pop off the repressor protein and cause induction. And then in scenario 2, you need the ligand to repress, so it needs to bind the repressor protein, and then removal of that ligand causes de-repression. Does that make sense?
The last one.
Yeah. So in scenario 1, the ligand is needed to pop off the repressor, to get the repressor off and then induce transcription. And then in scenario 2, the ligand is needed to repress the protein and repress the target gene from being expressed. And it's that removal of the ligand that we call de-repression, because then we're getting essentially induction by de-repressing.
So in scenario 1, we need the ligand to be able to express, and in scenario 2, we need it to go away and able to express.
Exactly, yep. And in scenario 2, you need the ligand to repress as well. Yeah, so this one's a little difficult because it's repression, but then there's these ligands that either allow it to induce transcription or de-repress transcription. So the more you go through it, it will make more sense. Okay, so now we're going to go talk about activators. Like I said, most activators bind poorly on their own, and they need an inducer ligand present. So here I'm showing you an activator protein in green and the ligand, which is the inducer here. So the inducer has to bind this activator protein, and when the intracellular concentration is high enough, it gets to bind and we get this expression of the target gene. And so when the concentration of this ligand is low, the activator protein is kicked off, and then the target gene is not expressed. So transcription stops. So this essentially works in the opposite way of what I talked about in scenario 2, where you need this ligand here as a co-repressor to repress, and here this ligand is an inducer for the activator. Does that kind of make sense? This one's more simple because most activator proteins need the ligand, so there isn't really many situations where the activator protein binds on its own. So there's really one scenario. Activator protein binds the ligand inducer, and it activates transcription, but then when the ligand comes off, that weakly bound activator protein also dissociates, and then you get transcription stopping. Make sense? Okay, so far we're talking about sensing what's going on inside the cell, right? We're talking about ligands and things like that the cell is sensing. But there are extracellular conditions in the external environment that also are triggering these changes in gene expression. So this is a very common mechanism that's used by all bacteria to collect information from the outside, and it relies on systems called this two-component signal transduction system to sense the external environment. So this will take a little bit of memorization as well when you're trying to think about when you're sensing the internal or external environment. We're talking about the external environment. We're going to talk about this two-component system with a sensor kinase and a response regulator. So here I'm showing you the sensor kinase. It's this protein that spans the membrane. And so this is the sensor kinase basically is responsible for binding to some signal. It's sensing something, so it has to have that environmental signal and detects that environmental signal from outside the cell. And so basically when it binds, it activates phosphorylation. So signal triggers autophosphorylation. That's just what happens. And then we have this other protein that is in the cytoplasm, and so this is this phosphorylated response regulator. So this response regulator then takes this phosphoryl group during autophosphorylation, and it basically becomes phosphorylated itself. And so in this form, it's then able to bind to the chromosome operator sequence and alter the transcription of genes. So then it could act as either an activator or a repressor inside the cell. So the previous few slides, I was just kind of explaining repressors and activators at this level, but there are things that have to happen at the outside of the cell to lead to that activator or repressor protein. So we're kind of working backwards a little bit in our understanding. And sensing the external environment requires a sensor kinase that has to be activated through the signal or a nutrient or something in the environment. It autophosphorylates, and so this response regulator is in the cytoplasm. It takes this phosphate from the kinase and allows it to bind to the DNA and either activate or repress the transcription. Does that make sense? Okay. Quick concept check. Transcription is prevented by blank and stimulated by blank. So lots of terms, but this one is pretty straightforward. Let me take a minute, think about it. Yep, it's E. Transcription is prevented by repressors and stimulated by activators, so it's not a trick question. It's pretty straightforward there. Okay. So, I have about 20-ish minutes. Okay, so let's take some of these principles now and apply them to some operons. So we're going to talk about operon control. So these two scientists, Jacques Monod and Francois Jacob, proposed that genes could be regulated. So basically, in 1961, they noticed that in E. coli, there's this enzyme that basically is used to consume a carbohydrate called lactose. And this enzyme was only produced when lactose was present. So contrary to glucose, for instance, enzymes for glucose catabolism, so breaking down glucose, are always present in the cell, and they're produced at high levels because glucose is usually the favored carbon source for cells because it provides the quickest source of energy and it can get right into glycolysis. So glucose goes right through glycolysis, easily accessible, and those enzymes are constantly expressed, so we call that constitutive. Whereas these French scientists recognized that enzymes to break down lactose were actually inducible, and they had to be induced when lactose was present. So this was a big finding that launched this field of gene regulation, and they won the Nobel Prize for it. But the lactose utilization operon, or this LAC operon, is the LAC-ZYA operon of E. coli. It was the first regulatory system described, and so we still use this in so many different research labs today. So let's kind of go through a little bit of the paradigm of this LAC operon. So on the left, you have lactose. It's a disaccharide, so it's two sugars, and those two sugars that it is made of is galactose and glucose. And so these blue dimers or disaccharides are the lactose, and so in here we have this membrane protein called a lactose permease. Basically, it allows lactose to permeate through the cell, and this we call LAC-Y. And so there's an enzyme in here in the cell called beta-galactosidase, or we call it LAC-Z on the operon. And this is the enzyme that subsequently will cleave lactose into its two components, galactose and glucose. So it splits it into those two. And so we want this to happen because, like I said, we want things to be fed into glucose metabolism, which is what the cell ideally wants. It wants things to get to the glucose step so that it can go straight into glycolysis. So when there's high levels of beta-galactosidase, you'll get this breakdown of lactose into galactose and glucose. And so in another scenario, when there are low levels of beta-galactosidase, LAC-Z, then you have to have lactose converted into another molecule called allolactose. So this conversion will become important later in the lecture, but it's important to know that when there's a lot of beta-galactosidase, this enzyme can then break down lactose into these, and then you can have glucose go straight into glycolysis. But when there isn't a lot of beta-galactosidase, you have to convert lactose into allolactose. So if there's no permease to get lactose in, there's no enzyme in here, and so you can't take advantage of this carbon source at all. And so here I'm just showing you what the lac operon looks like and how it's organized, and it basically it just involves the regulation of the lac operon in order to utilize lactose as the carbon and energy source for the cell. And so it's a simple regulatory circuit, and so the lac operon consists of lac, Z, Y, and A, but we don't really know much about the function of lac A in lactose catabolism, so the two that you should know are the lac Z and lac Y. The Z is the beta-galactosidase enzyme, and the lac Y is the permease. And so in the absence of lactose, this lac operon basically is transcribed at really, really low levels, and the main reason is when there's low lactose, the lac operon is being repressed. And the thing that is repressing it is this protein product from this regulatory gene called the lac-I repressor. So this whole area makes up the lac repressor, and it's situated immediately upstream of the lac operon, but it's transcribed by a different promoter. So when we have low lactose, this lac repressor is there to repress making lactose because we don't have any, or breaking down lactose because we don't have any. So in this scenario, we have absence of lactose, and so this lac-I can then bind to this region. So how does lac-I repress the lac operon? It forms this tetramer. So lac-I is this red circle, and so you have this 4 of them, and basically it forms a tetramer that binds to 2 different operator regions that you don't have to really know, but just know that it binds to its operon region, and basically this binding partially overlaps the lac operon promoter, and this tetramer basically binds and loops the DNA and physically prevents the RNA polymerase from getting onto its promoter and then transcribing the lac operon. So there's this partial overlap. So when there's this DNA loop, the RNA polymerase can't bind and prevents transcription of the lac operon. Does that make sense? When you have low levels of beta-galactosidase, so this lac-Z, but you have lactose, but you have low levels of this enzyme that's supposed to cleave it, then you have lactose converted to allolactose. So then the beta-galactosidase can cleave and rearrange the lactose to make this inducer molecule we're calling allolactose. And so allolactose binds to the lac-I repressor and reduces its affinity to the operator and then allows induction of the operon. Does that make sense? Okay. So usually in the environment, there's more than one carbon source, and so the next question the cell will ask is, which carbon source do I use, or can I use all of them at the same time, or do I use one of them at a time? And so, as I've said before, enzymes for glucose catabolism are always present, and they're produced at high levels because glucose is the favored carbon source, providing the quickest source of energy. It gets right into glycolysis. And so many carbohydrates, including lactose, have to be converted to glucose first before they can be used in glycolysis, which is not ideal. So what these cells do is a phenomenon called catabolite repression, where you have a more favorable catabolite like glucose that will prevent the expression of operons that enable the catabolism of another less favorable carbon source like lactose. So here we're looking at bacterial growth in a batch culture over time. So over time here on the x-axis and on the y-axis, we're looking at optical density, or OD. I briefly mentioned this when I talked about ways to measure bacterial cell density, and so this relies on absorbance. But what you should know is essentially optical density is the growth of the bacteria over time. And so you see here that both glucose and lactose are present in the media. And at first, glucose is utilized and these cells are growing. But then when the glucose is all used up, the cell runs out of glucose and the cells completely stop growing. You have this lag phase right here. So this is taking up some time, and the reason for this is because the cells are retooling their genetic program at this point and getting enough de-repression of the lac operon to start using this less preferred carbon source, lactose. So then the lactose is utilized, and then once that's utilized, all of it stops, and then it's retooling its genetic program again to utilize something else. So this biphasic curve of a culture growing on two carbon sources is called dioxic growth. Questions? Okay. So having glucose prevents lactose from getting into the cell. And so if lactose can't get into the cell, the lac operon can't be induced. And so here, basically I'm showing you this glucose transport system. You don't have to know the details of all of this, but basically what you need to know is that when glucose is present, it inhibits the lac-Y permease activity. So this is the permease protein that lets lactose in. So glucose can get in, so the lac-Y permease is inhibited. This is called inducer exclusion. And so, yeah, if lactose can't enter the cell, then the operon can't be induced. And when glucose is absent, then you can have lactose get into the cell, and then you can break down lactose. So glucose is what's excluding lactose from coming in when glucose is there, when it's present. Let's see, I think I want to go through a couple more things. Actually, no, we can stop here because this is a good stopping point, and I can continue on next week with the rest of it. I know I threw a lot at you. I hope that review worksheet was helpful. I know this unit, it's a little bit more complex. There's a lot more memorization and details, so yeah, please ask questions when you have them. And yeah, I will see you on Monday.
Yes.